Hi everybody, please allow me to introduce myself. I am Nermin Ələsgərova. I am the teaching assistant at the Department of Epizotology, Microbiology and Parasitology at the Faculty of Veterinary Medicine. Today I am going to present my first lecture to our students and all of our listeners. Firstly, I'd like to say that I wish a strong health and success all of our students throughout the academic year. The topic of our today's lecture is the classification, morphology, and the structure of the microorganisms. Firstly, I'd like to give some information about the microbiology. What is the microbiology? Microbiology is a scientific study, and microbiology is the most interesting lesson from my point of view. Microbiology is a science about the microorganism. But what is the microorganism? Microorganisms are the extremely small organisms. That's why it's impossible to observe them by the naked eye. For the observation of the microorganisms, we need some equipments, some lab equipments, especially the microscopes. And there are some type of the microscopes, electron microscopes, light microscopes, and another. For the microbiology lessons, especially microbiology lab lessons, we need light microscopes. On the light microscopes, it's easy to observe the microorganisms. Microorganisms consist of some type of the organisms, for instance, bacteria, rickettsia, prion, virus, microscopic fungi, chlamydia, and another type of the organisms. And throughout the microbiology lesson, we try to teach the important points of these small organisms. And I always say to our students, the microorganisms are the invisible enemy for us, for animals, and for the plants. And as I mentioned before, microbiology is a scientific study about the invisible organisms. For the observation of the microorganisms, we need some type of the equipment. And under microscopes, we can to observe the microorganisms, especially bacteria. And for the main purpose of the microbiology is to diagnose bacterial infectious diseases, the treatment of these infectious diseases, and the prevention measures against these diseases. And microbiology have their own some main fields. First one is the medical microbiology, veterinary microbiology, agricultural microbiology, industrial microbiology, and environmental microbiology. Firstly, the medical microbiology. Medical microbiology is the microbiology about the application of the some drugs or the medicine, the treatment of human infectious diseases, and the prevention methods against these diseases. In addition to it, the industrial microbiology about the science of some kind of industrial process that microorganisms play a vital role on this process. The fermentation process, the pasteurization process such as, and etc. Veterinary microbiology focus on the treatment of drugs, implementation of the bacterial, viral, or fungal diseases. And second one is the treatment of diseases and prevention measures of these diseases. Agricultural microbiology is the microbiology about the some agricultural process. So these microorganisms play a huge role on the agricultural process. So microbiology is one of the most interesting lessons, and I think that we especially focus on the infectious diseases. On this stage of my speech, I'd like to give some information about the diseases. Diseases are divided into two main groups, infectious diseases and non-infectious diseases. There are main differences between them. For instance, infectious diseases have their own incubation period, and this incubation period lasts sometimes a long time or a short time. It depends on the disease, it depends on the etiological agent or the infectious agent. And second one, infectious diseases have their own etiological agent, and these agents can be rickettsia, prion, virus, bacteria, microscopic fungi, and etc. And I think that infectious diseases is more dangerous diseases in comparison with non-infectious diseases, because throughout the non-infectious diseases it's impossible to observe that one person or another person can be infected another person or another animal. But throughout the infectious diseases, the infected process can be spread from this person to another person, or from this person to the another animal. And microorganisms can be infected human, animal, and even if the plants. And there are some type of the infectious diseases. We say these diseases, the zoonotic diseases. The main thing of these diseases influence on the both human and animal. And microbiology lesson especially focus on these important points. And as I mentioned before, there are some type of the microorganisms, and bacteria one of them. Bacteria cause some kind of infectious diseases, and these infectious diseases are called bacterial infectious diseases. Sometimes it's easy to prevent or treat the bacterial infectious diseases in comparison with another type of the microorganisms diseases. And bacteria have their own structure like another living organisms, like another microorganisms, and this is the main structure of the bacteria under microscope. And bacteria have their own both internal and external elements. All of them are very essential elements for the bacteria, for the bacterial lifestyle and bacterial living. And first one is the cytoplasm, second one is the plasmid, third one is the ribosome, this is the food granule, this is the mesosome, and plasma membrane, cell wall, capsule, and flagella. All of them are the main element of the bacteria, and it's impossible to imagine the bacteria without none of them. And these structural elements have their own functions on the bacteria. And for the maintenance of the bacterial lifestyle and the bacterial living cells, we try to protect some kind of the organisms. And I can say that lots of people think that all of the microorganisms are dangerous, but this thought is the wrong, because there are some type of the microorganisms, they are good for our health. For instance, some type of the microorganisms, even if they are inseparable part for our digestive tract, like E. coli or the Escherichia coli. And I think that we should be very grateful this microorganism, because E. coli or Escherichia coli and non-pathogenic Escherichia coli are the inseparable part of the both human and animal digestive tract, and there are some type of the bacteria, they are good, and they are good for our health. And in addition to it, there are some type of the microorganisms, they are pathogenic microorganisms. It means they cause some infectious diseases, human, animal, and another type of the living organisms. And but third part of the microorganisms, they are neutral microorganisms. They are not good or bad for our health or the animal health. And bacteria is a type of the microorganism, and I always say that bacteria have more sophisticated structural elements and more sophisticated morphology in comparison with the another type of the microorganisms. When I compare the bacteria, prime, or another type of the microorganism. But why? Because bacteria have more sophisticated structural element. For instance, bacteria have both DNA and RNA. You know, DNA is the main element of the microorganisms, because DNA carry the genetic informations, and the main function of the DNA is to carry or to transmit the genetic material or the genetic information from this generation to another generation. So DNA have a vital role on the bacterial structure, on the human, animal, even if the plant. But what is the RNA? RNA is the another type of the nucleic acid, and is the main function of the RNA is sometimes play a vital role on the transition process and sometimes play a vital role on the energy source of the microorganisms, especially the bacteria. And ribosome is this type of the structural element like the RNA. This is the flagella. Please pay special attention to the flagella because all of the microorganisms don't have the flagella. But what is the flagella? Flagella is a type of the movement organ. By the help of the flagella, microorganisms can be moved or attached. Why I say that move or attach? Because some type of the microorganisms, they are not moved. They can attach by the help of this kind of movement organs. And microorganisms can have one flagella, 2, 3 or more than flagella. So flagella is the main movement organ. And from the diagnostic issue, flagella is very essential because some type of the microorganisms have this kind of movement organs, others not. For instance, E. coli have their flagella, one type of the flagella, and this is very important for the diagnostic point of view. And plasma membrane, cell wall, capsule. Now I'll clarify all of them. And this is the main bacterial structure. First one is the cell wall. But what is the main function of the cell wall? The main function of the cell wall is to protect the bacteria from the inappropriate or the dangerous external factors or the conditions, because you know lots of factors can be influenced on the microorganisms, not only bacteria. I mean all of them microorganisms. And the main function of the cell wall is to protect the bacteria from the external factors, the influence of the external factors. And these external factors can be temperature, high temperature or highest temperature, low or the lowest temperature, because microorganisms are very different based on the influence on the temperature. Some type of the microorganisms prefer high or the highest temperature. For instance, mesophile microorganisms are not this group. Mesophile microorganisms prefer 37 or 38 degree. But there are some type of the microorganisms, we call them psychophile microorganisms. They prefer high or the highest temperature. This temperature is the appropriate for them. But another type of the microorganisms prefer low or the lowest temperature. And the appropriate temperature for them is the low or the lowest temperature. Second one, pH. Microorganisms need appropriate pH level. Otherwise, it's not easy to grow microorganisms at the lab condition. And next one can be the light, influence of the light, and last one is the lack of the nutrition. Like another organisms like us, plant or the animals, microorganisms need nutritional elements. Some nutritional elements are the essential for the growing of the bacteria at the lab condition or the outside the lab. And microorganisms prefer the nutritional elements like the protein, vitamin or another type of the thing. That's why it's very important for the microorganisms. And cell wall try to protect the bacteria from the dangerous condition of the outside. And second one is the capsule. And the main function of the capsule is to maintain the bacterial lifestyle and the maintain the bacteria. And this is the pili. By the help of the pili, microorganisms try to move or attach. As I mentioned before, the main function of the pili is the resemble us the function of the flagella, and this is the DNA. Try to carry the genetic information from this generation to another generation, and this is the three flagellas of the bacterial structure. And this is the another slide of the bacterial structure, and this is the cytoplasm, ribosome, and inclusion, plasmids, and plasmids especially are connected with the DNA, and DNA are connected with the plasmids. And this is the flagella and pyllis, capsule, cell wall. All of them are the main structural elements of the bacteria. And without the microscopes, we can't observe the microorganisms under microscopes because they are invisible. That's why we need some type of the lab equipment like the microscopes for the observation of the microorganisms. And for your information, it's easy to observe using the light microscope for the observation of the bacteria, but sometimes it's impossible to observe the flagella using the light microscopes. For this purpose, we try to obtain the electron microscopes for the observation of the flagella. And in addition to it, I can see that we especially focus on the microorganisms because microorganisms are the main cause the some infectious diseases. That's why we try to learn microorganisms by because they cause infectious diseases around the world. And unfortunately, every year around the world, microorganisms, infectious diseases resulted in millions deaths among the human and animals. And in addition to it, microorganisms, I said that sometimes can be neutral. They are not good or bad for our health, but what is the main function of these microorganisms? In nature, we need the biodiversity. These microorganisms have their own role on the biodiversity at the nature. So we need also this kind of microorganisms. But especially we should pay special attention to the pathogenic microorganisms because they are the causative agent of the infectious diseases. And infectious diseases are subdivided into some another groups like the bacterial infectious diseases, fungal infectious diseases, and etc. And there are lots of taxonomic criteria on this issue. And the bacterial cell structure connected with another things. For instance, the external factors, sometimes the internal factors. And there are some type of the microorganisms, they have some type of the antagonist relationship between them, and in this process they try to die each other the result of this process. That's why we should be very careful against these invisible organisms, invisible microorganisms. And like another type of the organisms, microorganisms need appropriate temperature, appropriate nutritional elements, and another things. And we try to give appropriate nutritional elements to the microorganisms at the lab condition for the obtaining the culture of the microorganisms. And this is the bacterial cell structure, and especially the external structure of the microorganisms are called the morphology. So morphology means the external structure of the microorganisms, and based on the morphology, microorganisms are divided into some groups. And this is the plasm membrane, the function of the plasm membrane. And plasm, this illustration showcase the plasm membrane. Plasm membrane have both outer membrane and inner membrane. Both of them try to protect or maintain the plasm membrane. And plasm membrane contains first one the nutrition or the nutritional elements, because plasm membrane are the responsible for the nutrition and nutritional elements. And second one is the maintain organized status. And next one is the selectively barriers. You know, there are some type of the barriers, especially influence on the microorganisms in a bad way. I mean this one, and the prevention of the movement, because some type of the microorganisms try to move, and there are some type of the microorganisms they move very rapidly, like E. coli, and this one is the respiration, like us and another type of the organisms, microorganisms respiration process, and the respiration process is the connected with the plasm membrane, and this one is the photosynthesis process, and plasm membrane play a huge role on the photosynthesis process, and last one is the producing lipids and cell wall components. And microorganisms need some type of the nutritional elements like lipids, like protein, vitamin, and cell wall components, because they maintain the bacterial lifestyle is connected with cell wall components directly. That's why we need them, and this process is connected with the plasma membrane. And this is the bacterial cytoplasm, the main structure of the bacterial cytoplasm. Bacteria have the most sophisticated structure and more sophisticated structural elements, and this is the another essential element of the bacteria. Bacteria cytoplasm, this is the DNA, and DNA is the carrier genetic information, especially by the help of the chromosome. And I think that our students remember lots of things about the chromosome from their school years, from their biology lessons, and I can see that microbiology is connected with another subjects like biology, like chemistry, biochemistry, physics, and another type of them subject, because there are some special connection between them. This is the flagella, and it's easy to observe the flagella under microscopes, and for this purpose we try to implement some type of the methods or the techniques for the detection of the flagella, the movement of the flagella or the attached of the microorganisms. This is the capsule. This capsule is belong to bacterial cytoplasm capsule, and this is the cell wall, the protection of the microorganisms, especially the bacterial structure. This is the pilus and plasmid DNA, and another type of the ribosomes, and ribosomes is the main energy source of the microorganisms. In addition to it, Golgi complex or the Golgi apparatus and ribosomes, mesosomes are the essential elements of the bacterial structure. And this is the some information about the cytoplasm. Please pay special attention to the structure of the cytoplasm. This is the structure of the cytoplasm under microscopes. And cytoplasm contains these important points. First one is the ribosomes. Ribosomes is connected with the cytoplasm, the main energy source. Every living organisms need energy to maintain their life, and bacteria also. This is the nucleotide and chromosome, and chromosome is the main element of the genetic point of view, and plasmids. And this is the bacterial DNA and plasmids. As I mentioned before, bacterial DNA and plasmids are connected with each other. And bacterial DNA are the plasmids play a vital role on the learning of the bacterial structure. And bacterial structure, I can say that more sophisticated in comparison with, for instance, virus, because a virus have the either DNA or RNA, unlike the bacteria. But bacteria have DNA, both DNA and RNA, the main structural elements. In addition to it, the learning of this process, the learning of the microorganisms, we need some type of the resource, and there are lots of scientists around the world and in our country, our national scientists. I always say that we should be very grateful to these scientists. For instance, Galileo Galilei, Robert Hooke, Ilya Ilyich Mechnikov, and Paul Ehrlich, Antoni van Leeuwenhoek, Louis Pasteur, and another scientists. And in addition to it, there are some scientists, they have own contribution to the development of the microbiology in our country. And in addition to it, even if sometimes scientists say that Antoni van Leeuwenhoek is the father of the microbiology, based on their huge contribution to the development of the microbiology. And microbiology, especially in the first time, focus on the detection of the microorganisms, because in those years it's not easy to detect or to observe the microorganisms without the microscopes or another type of the lab equipments. Especially the lab lessons of the microbiology, we need essential lab equipments and for the observation of the microorganisms. And for the observation of the microorganisms, we try to implement some methods or the technique, for instance, for the preparation of the semia. After this process, we try to observe the microorganisms. And this is some information about the plasmid. What is the plasmid? Plasmids are small, double-stranded and close the DNA. That can be isolated from the bacterial cells, even if sometimes we need the isolation of the bacteria from the culture media, from the diagnostic point of view, because there are some type of the microorganisms, they can produce the poor culture. And for the diagnostic some type of the infectious diseases, we try to isolate the poor culture of the microorganisms, and there are some type of the special techniques. We try to use them, and they replicate independently of the bacterial chromosome, and this is the bacterial DNA and plasmids. So plasmids have their own role on the bacterial structure. In addition to it, there are some differences between the bacterial structure or the structure of the another type of the microorganisms, and this process influence on the classification or the taxonomy of the microorganisms. There are lots of criteria on this issue. And some scientists say that microorganisms are divided into three main groups based on the infectious point of view, pathogenic, non-pathogenic, or the neutral microorganisms. And another type of the microorganisms says that we should microorganisms some type of the another group based on their morphology, their external structure and their structural elements. And so there are lots of criteria around the world and in our country. And the classification of the microbiology or the classification of the microorganisms help us to differentiate some kind of microorganisms to another type of the microorganisms. So the identification of the microorganisms, because some type of the microorganisms look like another type of the microorganisms from the external structure of the some external or the internal elements. And this is the anatomy of the plasmid. This is the gene you want interested, and this is the promotion process promoter, and this is the selection maker, and this is the antibiotic resistant. And there are some type of the external factors influence on the microorganisms, and I said that temperature, pH, lack of the nutrition or the nutritional elements, and antibiotic or the drug or the medicine resistance are the one of them, because some drugs or some medicines influence of the microorganisms in a bad way, and this is the main purpose of the implementation of the drugs or the medicine in the medical microbiology and in addition to veterinary microbiology, because we try to kill this invisible enemy, the microorganisms. That's why we should some type of the methods, the implementation of the appropriate drugs or the medicine, and antibiotic one of them. And I can say that antibiotic is the enemy of the microorganisms, but in addition to it, sometimes the implementation or the utilize the some type of the drugs or medicine, they can kill the microorganisms, but unfortunately sometimes they can be damage our body or the animal body. We should be very careful toward this issue. Sometimes they influence on this our digestive tract or our upper respiratory tract, our blood serum, even if another elements of the microorganisms. We can easily observe the microorganisms everywhere, inside our body, outside our body. even if under space conditions, sometimes we try to generate the artificial space conditions at the lab for the growing the microorganisms. Sometimes microorganisms need some this kind of is the rod-shaped microorganisms. Second one is the coma-shaped microorganisms. Third one is the round-shaped microorganisms. Under microscope, we try to identify the microorganisms based on their external structure. And rod-shaped, coma-shaped and round-shaped microorganisms. They are also subdivided into some groups. For instance, round-shaped microorganisms are called coccus or the cocci microorganisms, and they are subdivided into some main groups like monocoque, diplocoque, pneumocoque, staphylocoque, streptocoque, and another type of the coque. And they are also the main causative agent of the infectious diseases. For instance, diplocoque cause diplococcus in human and animals. In addition to it, staphylocoque cause staphylococcus in human and animals, and they are main causative agent of the infectious diseases. That's why we should be pay special attention to these microorganisms, because you know, in most cases, the health of the human depends on the health of the animals, because all of us need animal products like egg, like meat, and another type of the animal products. That's why in most cases our health depends on the health of the animal. And microorganisms influence on us, and like these microorganisms influence on the animals. And this is the main structure of the microorganisms. And I said that the second one is the rod-shaped microorganisms. Rod-shaped microorganisms are subdivided into three main groups: bacteria, bacil or the bacillus, and last one is the Clostridia. For instance, some type of the microorganisms have their own rod-shaped structure, rod-shaped morphology. And I always say that our students, for instance, Bacillus anthracis, it means these microorganisms or this causative agent is the rod-shaped microorganisms, or Clostridia, Clostridium botulinum, or Clostridium tetani, Clostridium shawei, and it means that this kind of microorganisms are the rod-shaped microorganisms, and this process assists us to differentiate or to identify the microorganisms based on the morphology, based on the external shape of them. And there is another type of rod-shaped microorganisms, for instance, E. coli or the Escherichia coli, and there are some type of the round-shaped or the cocci-shaped microorganisms, monocoque, diplocoque, pneumocoque, and another type of them. And in addition to it is the third group is the coma-shaped microorganisms, and coma-shaped microorganisms also are divided into some main groups. For instance, they can be coma-shaped, just coma-shaped microorganisms. It can be spirillum, spirochete, and another type of the shaped microorganisms. And why we are interested in the shapes of the microorganisms? The main purpose of this issue is to diagnose the infectious diseases, because microorganisms are the causative agent of the infectious diseases, and we try to diagnose the infectious diseases using the shapes of the microorganisms, because the shapes of the microorganisms are different, and we try to using the shapes of the microorganism to diagnose infectious diseases. And this is, for instance, Clostridium tetani. It means this is the rod-shaped microorganisms. In addition to it, Clostridium tetani have their own survival mechanism, like the spore, because this kind of survival mechanism help the microorganisms to maintain their lives. And this is another type of the microorganism, and for instance, Salmonella species or the Brucella species, all of them have the different morphology or the different external shape. And this is the Clostridium botulinum. Clostridium botulinum is the one of the most dangerous infectious diseases causative agent. It called the botulismus. And in addition to it, another type of the microorganisms, and these are the cocky-shaped microorganisms. And cocky-shaped microorganisms or the round-shaped microorganisms can be observed like chain or the grape on the microscope. And this is the another shapes of the microorganism, coccus-shaped microorganisms or the round-shaped microorganisms. This is the bacillus, rod-shaped microorganisms, and there are lots of rod-shaped microorganisms, for instance, Bacillus anthracis or the Bacillus cereus. But what is the main difference between them? The main difference, for instance, between the Bacillus anthracis and the Bacillus cereus, not their external shape, because their external shape is the same. Both of them have their own rod-shaped microorganisms. They are the bacillus. The main differences between them are the response to the some type of the drugs, for instance antibiotic, or the another type of the external factors. In addition to it, Bacillus anthracis have the survival mechanism. This is the main differences between them. So I try to say that sometimes the shape of the microorganisms can't help to diagnose or the identification of the microorganisms. And this is the streptococci. Streptococci resemble the chain, and they observe like the chain on the microscope. And this is the diplococci pairs, two cocci types. And this is the spirochete. This is the coma-shaped microorganisms, like for instance Vibrio cholera. This is the third type of the microorganisms. And in addition to it, sometimes we need another type of the requirements for the growing the microorganisms. Microorganisms need some nutritional elements or the nutrition at the lab condition, and this purpose we try to give appropriate nutritional elements to the microorganisms, for instance to the bacteria or the microscopic fungi, because for the growing and developing process, microorganisms need growth mediums. But what is the growth medium? Growth medium is a type of the growth medium. It's easy to grow microorganisms on them because this is the appropriate environment for the microorganisms. And there are three types of the growth mediums based on their physical states: solid, semi-solid, and liquid growth mediums. And growth mediums can be artificial and natural growth mediums. Natural growth mediums can be blood serum, milk, meat, egg, and another type of the nutritional elements or the products, especially animal products. We very easily use the animal products for the preparation of the growth mediums. Second one is the artificial growth mediums. What is the artificial growth mediums? Artificial growth mediums is a type of the growth medium is using for the growing the special purpose microorganisms. There are some type of the microorganisms, they are very selective for the growth mediums, and in these cases we should prepare the artificial growth medium at the lab condition. And artificial growth medium can be selective or the differential growth mediums, because like the Brucella, Salmonella or another type of the microorganisms are very selective to their growth mediums. That's why we need some type of the microorganisms based on the growth mediums. And growth mediums should contain appropriate nutritional elements, protein, vitamins, some microelements, because microorganisms need all of them. And next one is the appropriate pH level. Microorganisms need the neutral pH level, because this is the appropriate pH level for the growing microorganisms. And last one is the microorganisms need appropriate temperature for the growing process, and they are very different based on the temperature. They prefer different temperatures for the growing or the development of them at the lab condition. And this is the survival mechanism of the microorganisms. Survival mechanism of the microorganisms are connected with their morphology, because this is the external shape of the microorganisms. Some type of the microorganisms have this kind of survival mechanism. Survival mechanism can be spore, endospore or the capsule. Under inappropriate or the bad environmental condition, microorganisms, some type of the microorganisms try to generate their survival mechanism like spore, endospore, and Bacillus anthracis is this type of the microorganisms, because Bacillus anthracis have the ability to generate endospore under inappropriate environmental conditions, especially on the soil. That's why scientists say that this causative agent is the soil microorganism. And next one is the capsule. Inside our body, inside the human body or animal body, some type of the microorganisms have this kind of ability to generate capsule. Also, capsule is one of the survival mechanism, and this is the main structure of the Bacillus anthracis spore. And this is the DNA and cell membrane, cell wall, capsule, spore membrane, spore wall, spore DNA, spore coat, ribosomes, cytoplasm, cortex, and endospore. And this is the Bacillus anthracis rods, rod-shaped microorganisms, and this is the spore, the survival mechanism of the microorganisms. Using some type of the staining methods, we try to observe the capsule or the endospores or spores of the microorganisms. And this is the another survival mechanism of the microorganisms, and we can observe them, these survival mechanisms, under microscopes using some staining method. And this staining method is the special staining methods. And this is the survival mechanism of the microorganisms. This is the vulnerable anthrax, and this is the protected anthrax. Using the survival mechanism, bacteria or another type of the microorganism like Bacillus anthracis try to protect themselves from the inappropriate or the bad environmental conditions. And on this stage of my speech, I'd like to give some information about the staining methods, especially the gram stain, based on the morphology of the microorganisms. For the observation of the microorganisms, we need some type of the staining methods. And these staining methods are divided into two main groups. First one is the monochrome staining methods. Second one is the more sophisticated staining methods. And scientists consider that it is not useful using the monochrome staining methods. But why? Because scientists think that using the monochrome staining methods, it's impossible to observe the microorganisms and to identify the external shape of them. For instance, some of them are the Coma shaped, the rest of them are the rod shaped microorganisms. Using these methods, it's not easy to observe them under microscopes exactly. That's why we should use the more sophisticated staining methods, and there are lots of more sophisticated staining methods like gram staining or the Sil-Nielsen, for instance, staining methods, Romanovsky-Gimza, and another type of the staining methods. And gram stain or the gram staining method is the one of appropriate methods on the microbiology lessons. And firstly, we need some uncontaminated or the sterile slide, and of course we need some type of the samples. These samples can be blood, serum, milk, even if these samples can be the some type of the culture media. All of them are considered the appropriate samples for the preparation of the simia. Firstly, we should prepare the simia, and after the preparation of the simia, we try to fix or the fixation process of our slide. And there are two types of the fixation: chemical fixation and physical fixation. For the purpose of the physical fixation, we use the lamps, special gas lamps on the lab condition, and for the purpose of the chemical fixation, we need alcohol. And in addition to it, the preparation process of the simia process, we need some type of the stains, and these stains should be the neutral stains. We use the neutral stains. They can be Lugol solution, Pfeffer solution, and Romanovsky-Gimza stain, or another type of the crystal violet, or another type of the neutral stains, because microorganisms prefer the neutral pH level. In other cases, microorganisms can be change their external shape, or they can be change their morphology, and we need to differentiate microorganisms exactly. That's why we should use this kind of neutral stains. In addition to it, based on the gram staining, microorganisms are two types: gram-positive microorganisms and gram-negative microorganisms. Gram-positive and gram-negative microorganisms, lots of bacteria or the another type of the microorganisms can be gram-negative or the gram-positive. Please pay special attention to this point, especially peptidoglycan. First one, gram-negative microorganisms. Gram-negative microorganisms have just one chain of the peptidoglycan, but unlike it, gram-positive microorganisms have the 1, 2, 3, and 4 chain of the peptidoglycan. So the main purpose of the gram staining, the process is connected with the peptidoglycan. The number of the peptidoglycan chain. That's why microorganisms can be gram-negative and can be gram-positive, pink, or the another color, and everything depends on the peptidoglycan and their soluble. And in addition to it, gram-negative microorganisms are the outer membrane, unlike the gram-positive microorganisms, because gram-positive microorganisms not the outer membrane. In addition to it, and gram-negative microorganisms have their own special structure, unlike the gram-positive microorganisms. And in addition to it, gram staining methods are named after the Christian Gram, Hans Christian gram, and we should be very grateful to these scientists for the gram staining methods. And this is the results of the gram staining. For the purpose of the gram stainings, there are two types of the result: gram negative and gram positive microorganisms. Some type of the microorganisms are gram negative, the rest of them are gram positive. But there are some type of the microorganisms, it's impossible to use them for the gram staining methods, like the mycobacteria and another type of the microorganisms. In this purpose, we try to prepare another type of the staining methods, for instance, Steele-Nielsen methods or another type of the staining methods. And the survival mechanism of the microorganisms, like capsule, spore or endospore, also it's easy to stain them using appropriate staining methods. And the staining methods always implemented at the microbiology lab for the observation and identification of the microorganisms. Otherwise, it's not easy to observe the microorganisms, even if it's easy to observe the movement of the microorganisms. And gram-negative and gram-positive microorganisms, these are the main differences between them. They are very different in comparison with each other, especially for the peptidoglycan and outer membrane issue. Outer membrane is a type of the barrier for the gram-negative microorganisms. The first stain of the gram staining process is the violet, and gram-negative microorganisms not penetrated to this type of the stain because outer membrane some type of the barrier for this purpose. But gram-positive microorganisms is a type of the easy microorganisms from this point of view because they don't have outer membrane. That's why all of the stains try to penetrate the gram-positive microorganisms. That's why the color of them are different. And in addition to it, we at the last point of the staining process, we add immersion oil to our semia. But why? The main purpose of the utilize the immersion oil is the to identification and to obtain the clear view on the microscope. That's why we should add the immersion oil. Criteria, or they have different taxonomy on their classification. In addition to it, sometimes we need another type of the classification of the microorganisms. So I can say that we should pay special attention to the detection and identification of the microorganisms because they are invisible enemy and they influence on both human and animal. We should be very careful against them and against the influence on them. So I think that our students should learn microbiology very well, otherwise it's not easy to protect us, our loved ones, or our animals. As I mentioned before, in most cases our health depends on the animal health. So I think that microbiology is one of the most interesting subject, and this is the end of my today's lecture. This is the another gram-positive bacil. Thank you so much for your attention. Thank you so much for listening to me. Thank you. Is there any additional question?